right now. So thank you so much for attending. I basically just want to give an introduction and then I'll introduce the special speakers here today. But I did want to say that today is the second year we're hosting the expert webinar series, and we are very excited to share with the industry some of our groundbreaking variable path length applications being implemented across the world. The whole purpose of the series is to educate the biologics industry about variable path length technology and how it could be implemented as an inline analytical measurement to provide real-time monitoring, better driven control, and minimize risk during the manufacturing process. Process analytics technology is a hot topic in our industry, but it takes strategic planning and coordination to properly implement concentration measurements as part of PAT strategy. We will bring together some of the industry's true pioneers in PAT implementation to share with us the process they took to implement the FlowVPE as part of their PAT strategy, with various steps in the workflow. My name is Brandon Goldberg. I'm the Senior Field Application Scientist, and I've been with Repligen for over 6 years. I'm very excited to see where this technology started and where it's going right now, and how our customers are applying it to their processes. However, today we have the pleasure of introducing two experts from Janssen Pharmaceutical, Marius Mueller, the team lead analytical fill finish support, and Renata Positarenko, the associate scientist. So first, I will start off a little bit about Marius. He leads the analytical fill finish support team at Janssen Schaffhausen, Switzerland. His team is responsible for analytical support study, as well as scouting, development, and implementation of the new PAT tools for drug product manufacturing. Marius joined Janssen as a postdoc in 2018, establishing new analytical technologies for protein stability and aggregation. He holds a PhD in a field of mammalian synthetic biology and metabolic engineering from ETH Zurich in Switzerland. Prior, he received his master's from the University of Strasbourg in France as part of a tri-national study program. For Renata, an associate scientist at Janssen, she received her bachelor's and master's in science in chemical and process engineering from the University of Padova, Italy, in 2019 and 2021, respectively, focusing on process system engineering, chemometrics, and statistical monitoring. During her master's, she spent 3 semesters traveling abroad during in Germany, Belgium, and at Janssen, focusing machine learning and simulations. She is passionate about mechanistic and data-driven modeling as tools to enhance and optimize pharmaceutical industry processes. Sorry about that. So just as a reminder before we get started today, you may enter questions in the question box to your right during the control panel, and there's basically a whole tab for you to enter your questions, and at the end of the presentation, I will read off all the questions that people have during the QA session, and the speakers will be able to answer your questions then. However, once they get started, we would like not to interrupt them. So today, let's get started with the presentation, the automation of biologics drug product manufacturing through modeling and FlowVPE. Marius and Renata, thank you for joining us here, and please take it away, Marius.
Thanks so much, Brandon, and good afternoon, good morning, good evening, wherever you are, and yeah, welcome to the talk that Renata and I will be giving. As Brandon said, we will talk about the model predictive control for automated biologics drug product manufacturing. So we will basically really start off, or I will start off with showing you on where we started, a bit also on the whole history, and then hand over to Renata for a bit more on the technical details and really how we established ultimate process control. Let me start off with an expression where it says that the best sample you take is the one you're not taking. And in this regard, basically what we have in Schaffhausen, that's basically also a bit on the strategy that we are following within whole Janssen. In order to do so, not taking any sample, what we would do is really to go for online analytics, to really bring the analytics we're currently doing in the lab to an online stage, which would then eventually result in real-time data that is generated that we can then either use for process monitoring, for better process understanding. But eventually, and that's really where we are heading with the whole strategy, is to really then go combine it with an eventual scientific model to use it, even then control the process and lead then to a certain feedback. This overall would then really allow us to create, first of all, data, use that data then in a certain fashion, and then really even automatically work with the data generated. Let me start off here with the example of, and that is basically where we have been collaborating quite a long time, is using the FlowVPE for online protein concentration measurements. Here depicted is the drug product fill finish process that we have over in Schaffhausen, right? And what you can see is on the left, and let me just guide you through this quickly, is on the one hand, you have the biotainer with a certain drug substance. We do have also then the buffer tank, which then includes excipients and water, which is then diluted in the compounding tank. And then usually via a certain filter set is then resulting in the surge tank, which then ultimately is then used via the filling pump into the dedicated vials. Sounds actually pretty easy, but if you look at it a bit more closely, you can see there are several points in this where really protein concentration plays a major role and is critical. So it's on the one hand definitely also on the incoming drug substance. It is during the dilution process, and eventually it's for the final vial that is filled and sent for release. So we went out to say, okay, this is where we want to be, where we basically want to establish a potential online solution, but at the time we didn't have anything out there. So we went out and did this thorough technology due diligence. And we scouted for the full spectrum, so to say, including UV, NIR, Raman, as well as even terahertz technologies. The parameters we used for scouting included, on the one hand, accuracy and precision to really see that we are as good as or even superior to our offline methods used. We also had to include specificity to see that the background used in our product filling does not have any impact on the readings at all. And then last but not least, also a certain implementability together with engineering, together with regulatory, together with QC, to end up with a potential solution that would actually allow for all of this. And surprisingly, our selection fell for the FlowVPE system back then, where we saw the online potential as technology-wise, it is similar to the offline technology currently used with A280 rather than in an online fashion. So having found this, we then stepped further, and that basically was when the journey started in 2016. And here we basically had to start, as I just said, on implementability. We also started, first of all, having a certain cleaning assessment also done. And we actually found that was basically working closely together with C Technologies Repligen back then to find a solution for also autoclavability of the flow cells, where we had seen that the initial design did not withstand several autoclaving cycles. So hence we had several iterations to come up with a final viable solution to really use that in a GMP manufacturing. And indeed, in 2016, we were one of the first companies to use the FlowVPE in a GMP environment, which you can see here on the right-hand side. Maybe some of you might also look a bit weird on this, on the card we are having. So that was really like some of the first build-ups we have. Now the cards are much more sophisticated, much more having a much smaller footprint. But back then, that was basically how the setup looked like. And you can see all the connections, pump, and so forth that we had back then. Fast forward to today. So as mentioned, this would all not be possible with the close partnership that we have with C Technologies, which included several iterations on several hardware as well as software. We use it for several stages. So that's basically also a bit on how we are dealing with it. So we would first start with monitor the process to get a more in-depth process understanding. To then eventually, when having enough gathered of data, we would then use it for a potential replacement of IPC. to eventually even release replacement, to then, and that is basically also the core of this talk, is then to have really a controlled dilution and really use the data in a PAT, in a process analytical fashion, to feed back the readings to certain actions. Also, the field now has also broadened quite a bit. So starting off with drug product for compounding, we now even see this as a use for potential steps during UFDF steps for drug substance, as well as also tighter determination for our vaccines colleagues. Jumping a bit more closer, and I really want to emphasize this here as well, is that without this collaboration, I think we wouldn't have been able to do so. And one of this was basically also to be part in the FlowVPE beta testing. We really had also people from the shop floor, and we really had analysts, engineers to demo like of the first beta systems, where we could really then also in close collaboration and partnership, I would call it, have feedbacks on usability, on software and so forth. And that is also true not only for hardware, but also software, where we really are also closely working for that in order to really have a qualified system, where we also employed regulatory, where we also employed QC in order to see how to really get the system fully qualified and GMP compliant in that sense. Lastly, so we mentioned to roll it out to now more than 5 sites, and still counting. Amongst those are several in Europe, but also over in the US. And also quite a highlight is that we now have this included in several INDs and BLAs already, having FlowVPE in there. So again, just also highlighting once more quickly is, so we use it on the one hand really also together with CMOs, where we're really applying these technologies. We have it also for R&D labs, where it comes back to the process understanding for several of these steps. We use it for vaccines and several vaccine production sites. And then the 2 big ones are really like drug substance at several stages where it's applied, mentioned UFDF, but last also drug product finish site in Schaffhausen. Let me also jump a bit closer to this one. Also, they have several kind of applications in the fill finish surrounding. And as you might recall from the beginning, when I showed you the map of the compounding stage, there's even more to this where we can use it. So one of the first application could also be, as you can see here on the left-hand side, is filter absorption, where we can then really see the step, how much of the product is basically absorbed by a certain filter. And as we currently have it, it's you would rather measure before and after a certain mixing time to ensure that that enough material is basically or like the filter is basically saturated. So that was usually done by just saying a certain time. What we now have, and this is for the first time, also the applicability and the use case, where we can really directly show at what time the filter is saturated and how much material basically only needs to be avoided. The second step is then directly at the filling. So due to the real-time status of the FlowVPE, basically we can really enable to have even kind of a fingerprinting of each vial, and even knowing the concentration at each of the time that during a certain fill has a certain vial has. And lastly, and this is the one where then also we'll come in for the second part of the talk, is then really use it also for dilution. This is then really to have an automated dilution process. Again, here, what we currently do is using weight-based dilution process, where we basically calculate by weight how much diluent needs to be added currently. However, by really going for the CQA and the critical quality attribute, which would be protein concentration, one could really then directly follow and really control the process neatly in order to get the final concentration that is basically required. With this, Renata, I would like to hand over for how we basically got from this manual part to the automated dilution.
Thank you, Marius. Good morning, good afternoon, everyone. So I will guide you through the principles of the algorithm we built for dealing with the automation of the dilution process, as Marius anticipated. So before going into the details of the algorithm, I would like to bring you through how the dilution is currently done, and then through how we envision it for the future with our automation vision. So currently we have two tanks, one for the drug substance and one for the buffer. And as Marius anticipated, there is an operator that takes samples first from the drug substance tank, brings them to the lab for the measurements, for the offline measurements. Based on the results from these measurements, he calculates the amount of buffer to add, and then he adds it into the drug product tank. And with that, usually the dilution is done in 2 steps for safety reasons. So first, a certain percentage of the buffer is added in the drug product tank. And after this addition, the tank is sampled again for another measurement and another calculation, and then the second part of buffer is then added. So as you may notice, this is an articulated process to reach the final target concentration, and being so, it may be affected by inaccuracies and errors along the path. So and together with that, a disadvantage of this procedure is that we don't really know what is going on in our drug product tank. We only have some snapshots at the beginning before the dilution. So we have the knowledge of the drug substance concentration, and we have some other few measurements of the drug product concentration, but we don't know how the concentration is changing in our drug product tank while we are adding the buffer. So from that, we are thinking to improve this procedure through our automated procedure. So with that structure of our setup would change quite a little bit. So instead of having an operator that makes the calculations to add the right amount of buffer into the drug product tank, we would have now some online measurements provided from the FlowVPE and an algorithm that does these calculations based on the measurements from the FlowVPE. So now, as you may notice, the human intervention is removed. We don't need anymore that someone makes the calculations to find the amount of buffer to add, and we don't even need anyone to sample the tanks or the reactors and perform the measurements in the lab, because we can do that with the FlowVPE and with our algorithm. And the advantage of this new setup and new structure of the dilution process would be that we also have the knowledge in real time of how the concentration changes. And this is fundamental to calculate the right amounts of buffer, but also to intervene in time if there are deviations from the expected behavior, so that the algorithm can take the right decisions to bring the system to the final target concentration as desired, even though unexpected behaviors or unexpected deviations occur. So now that we understand which are the main ideas and the main drivers behind the need to automate the dilution process, we can deep dive into the technical aspects of the algorithm. So first of all, let's have a look at the overall workflow, or better, of how the algorithm relates to the process. So the algorithm is represented by the estimator and the optimizer, and these two parts communicate with the process through the input that can be the input for a pump that controls the dosing of the buffer, or for a valve, it doesn't matter, and through the outcome, that is the measurement from the FlowVPE. So in our workflow, the estimator uses the measurements from the FlowVPE to calculate the real state of the system, and we will understand why we need that in a moment. And this real true state of the system is then used in the optimizer to calculate the optimal input to give to the process in order to bring it a step closer to our set point and to our desired final concentration. In our estimator, we use both a mechanistic model and a data-driven model. And in our optimizer, we use again our mechanistic model of the system, a cost function that weights both how far we are from our target concentration and which are our ideal inputs to bring the system to the set point. And through these two contributions, together with some constraints, the optimizer is able to calculate the ideal trajectory to the set point, as we will see in a moment. Okay, so let's have a look at why we need the estimator. So the need for an estimator comes from the fact that there is one main complexity in the dilution process that is related to the time delay that we have. And the time delay the time delay is the time we need to wait in order to see from the measurements the effect of the dilution step that we have performed. And we can understand this a bit better by looking at the graph on this slide. So let's think that we start our dilution at time 0, and we add a certain amount of buffer. And let's say that at 100 seconds, we have a measurement from the FlowVPE. And this measurement, that is around 8 milligrams per milliliter, would be what the FlowVPE says our concentration in the system is. But we need to be careful about interpreting this measurement because it takes time for two fluids to mix together and to reach an homogeneous state where we can be sure that what we have added is perfectly integrated into the system, and the system is perfectly homogeneous, so that the concentration is the final concentration that we can expect as a result of what we have added as an amount of diluent. And for this reason, we cannot use this 8 milligrams milliliter measurement as it is in our optimization, because that would lead us to an over dilution in the end, and so we would cross our target concentration. We need to infer from this measurement what our real state in the system will be when the mixing would be complete. And with that, we can think that our real state is something around 5 milligrams per milliliter on the red line in correspondence of 100 seconds. So now that we have the real state of the system, we can use it in the optimization. And the optimization is performed according to the theory behind of the model predictive control. So we have our real state of the system, and we want to predict how the behavior of the system would be in the future given our current state. So we perform the optimization, exploiting our model of the system and giving the constraints we need in order to direct the optimization towards the right direction. We predict the ideal path, the ideal trajectory towards the target concentration that we want to achieve in the end. And based on this ideal trajectory, we obtain a series of optimal inputs that we should give to the system in order to reach our target concentration with this ideal trajectory. We perform this optimization at every measurement available, because at every measurement available we have more information, more knowledge about our system, and we can make better predictions on the future. So at every measurement available, this optimization is reperformed, and only the first action in this ideal series of actions is implemented at each measurement available. So now that we know how the real state of the system is calculated, is obtained, how the optimization is performed, there is one last important, sorry. I'm sorry. So now that we know all these things, there is one important thing for our feedback control loop, that is the tuning of our parameters. And we have tuned our parameters using some simulated experiments. So we have explored the impact of our tuning parameters with simulated experiments. And in this slide, I will show to you how which is the impact of two very different sets of tuning parameters on our dilution. So before we start these animations, you can expect to see in the upper plots the change of the model concentration with the red line, and of the measurements or what we can expect from the FlowVPE. with the dark dots, and on the lower plots we will see how the ideal inputs would change, or which are the ideal inputs that our algorithm would calculate at each measurement available. Okay, so we see now on the left-hand side how a dilution with more efficient parameters, and on the right-hand side the same dilution with less efficient parameters. But what do we exactly mean with less efficient parameters? Well, it's just a more conservative approach that would lead us to a slower dilution. So the important thing to notice here is that we have a lot of room to play with our tuning parameters. The algorithm, in the way we built it, is quite robust and can handle a wide range of tuning parameters, and we can choose them in regards of what system we have. So, for example, with more critical systems, more difficult systems, for example, where we have difficult fluid properties, we may want to use less efficient parameters or more conservative parameters. And with that, I will quickly show to you how we have tested our algorithm. So to test the algorithm, we have built a workflow of actions that orchestrates every action we need to perform in order to be able to do the dilution in an automatic way. So in our workflow, we are able to take the input variables, the receipt of what we want to perform, so which is our target concentration and a few other information that we need to know about the system in order to perform this dilution. And then we also communicate with the FlowVPE, and we take the measurements from it. We run our algorithm, and we write the ideal set points to our dosing element that can be a pump or a valve. And with that setup, we have tested quite some conditions to see how different geometries or different volumes or different fluid properties impact our algorithm. And we have obtained that the algorithm is robust towards these variations that we have included in our setup. And the last thing I would like to say on that is that we tried to reach our target concentration with a quite tight acceptance criterion that is plus minus 2% from the target. And we were able to achieve this because we have a robust algorithm, but also, and I would say more importantly, because the FlowVPE allows for this high precision and very good measurements. So with that, I am at the end of my presentation, and I would like you to remember a few key messages from what Marius and I presented today. So the main important thing is that it's possible to build an end-to-end process automation thanks to the FlowVPE. We did it. We can reach the target concentration based on the measurements from the FlowVPE. The FlowVPE allows us to remove the samplings from our tanks and from our reactors, and allows to remove the human intervention in the process when concentration measurements are needed. And we can save a lot of time with the automation of the dilution process. Thank you very much for your attention, and I would like to thank all the people that contributed to the realization of this project.
Thank you so much. So everyone, that was the end of the presentation. So thank you much for joining, and thank you, Marius and Renata, for presenting. We are now going to jump into our Q&A session. Just a reminder, if you have a question for today's experts, please enter your question in the control panel in the question tab. Okay, so let's jump right in, since I do see some questions coming in already. Just want to make sure. Okay, so I will start with this one. What new insights has the FlowVPX provided into your process since your team has incorporated it, either in PD or now?
Okay, Renata, I can start with this one, right? So as I've shown, right, so we used it in actually quite a broad field and broad variability in applications. And to start off with something like the filter absorption, that is really something that we couldn't see before. So usually due to just also the limitation of sampling, that was just not possible. We now actually know on how much sample is basically really recovered by, or basically absorbed by a filter over before, and even like shorten this time. But that's just like one of the few examples where there's really a great use. On top of it, we also now really have a certain process understanding that we get also now in terms of how long does it take, when do we have an homogeneous solution, for instance, for the mixing. That is also things that are really now just possible with really having this online analytics. And also just recently when we established this also, as mentioned with our drug substance colleagues, was also for the UFDF steps, where now it's even possible to really observe like up concentrating of material, how long it takes, and really get a characterization of the process, which also has not been possible before.
Nice. So how much, you mentioned that it saved you guys a lot of time. Did you say how much time or overall do you think you save from this? So by using the FlowVPX, sorry.
Mm-hmm. Yeah, I can take that one, Marius. Currently the dilution process with all the manual interventions takes from 6 to 8 hours to perform on average, and by avoiding this sampling and the measurements in the lab, we would only need the time that is required for the dilution to be performed. So this depends on, in the end, only on the process characteristics. So the tubing size, the amount of volume to add. So, just about, it depends only on how much we need to add and which is the capacity of the process. So with ideal conditions, that would be reduced to 1 hour, we can say.
Thank you. So one question that just came in is it very difficult to introduce this measurement into the product regarding the filling of the production?
Well, yeah, I wouldn't say it's easy, no, not at all. So it's really something that, as you saw, I mean, starting off in 2016, and we just basically had quite a journey to get it in there. So there were really kind of several pitfalls we had to take, which would also include convincing of regulatory, right, to bring this in. Also seeing on how much would be then the difference, and that's always what we have to show, that we are at least, and that's what I also mentioned, to be at least as good as the current offline technology. So that is really also from an analytical perspective. but then also having a certain paradigm shift to really also tell people, okay, we are moving the analytics to the line, whereas it's not done anymore in the lab, but it's more something that even an eventual operator has to perform. An operator has to set it up, and now even the operator basically will start this analytics, whereas before it was really just sampling, and then everything else had been done in the lab. So this is really also a bit of the paradigm shift that came with it, and that basically we had to take over the time, also educate, which is really just on top of all the implementation work that we did as shown here.
Okay, thank you. So let's see, another question is, there's a big push on automation and Pharma 4.0. How do you see the technology like the FlowVPX fit in the concept, and what improvements would you like to see realize the concept of Pharma 4.0?
Yeah, also a good one. So that's really, as mentioned, so that is really also the strategy generally we are following to really go into having it more automized, having it as shown with this great example to really be able to control the process. What we have seen during this implementation is also the link with software and the digital link that is basically required to really also move on data that is generated that basically directly gets into a cloud system that basically also complies, where really the data can then be used, and eventually really with the model that Renata showed to directly be used in real time to really control the process. So this is really something where I would also see that from the digital side, that this is really something to come up with, and also like the orchestration that we did, right? So that's also really some key element in the whole thing.
Perfect. Okay. So what challenges do you foresee when transferring to a commercial drug product manufacturing?
We can also take this one. So we did this, we already have it in place for commercial for some products. We did this transfer also over time. The challenge really moving it from lab scale to full scale is really to see also the comparability of how can you move things that you usually would perform in a lab that you basically have to something that is really on a several hundred liters of scale process. So this is really something where we also needed to really talk a lot with regulatory back and forth, talk a lot with sites to how much can we leverage, how can we close gaps, and how can we mitigate that. But overall, I guess we have been successful there. So yes, it is used in commercial, but eventually the next step would really also be for controlling the process, and I think this is still to be seen. Or anything to add there, Renata, on the commercialization part?
Not really. You explained it very well. So yeah, as the next steps of our automation work, we are now working on developing the infrastructure capable, the infrastructure in terms of data capable of hosting our algorithm. So we will see how it goes. It's all work in progress, but we are doing great.
Okay. So I think there are a few more coming in. So for the MPC, what reference / resources were used to build the final mode? How much retrospective data was needed to validate the models?
Mm-hmm. So as said, we are not yet at the validation phase. So yeah. Instead, for the reference material here, I would say that I'd like to acknowledge the help from colleagues from ETH Zurich. They helped in building the algorithm and to understand the difficulties of our process and to successfully deal with that complexity.
Okay, thank you. So I know we talked about time, so I think this question is going against following up with that. But they mentioned, how much time will this MPC/automation method save Janssen? And the second part of that question is, have you quantified the improvement of efficiency time saved in dollars?
So again, we are not yet at that stage of the project, so we don't have for the moment estimates for the savings in dollars. What we have though is that the automation of the dilution process would help with saving a lot of time and remove a lot of human intervention, and therefore to reduce the risk related to human intervention in the process.
Okay, nice. So, and I guess this is more of a question for myself, but I know working with the FlowVPX, it's usually pretty easy to add into the process, but I guess it's the change of the process that takes time. Is that correct?
So yes, I would say both, right? So it's really like also get it from, and I mean also speaking from the FlowVPE, right? As mentioned, so that would be really something where you say, if you don't have analysts also on the shop floor, right, it's really something to educate, to get them there. And I think that was also a great exercise we did with the FlowVPX, really to get it as lean as possible. Also, when you're comparing the construction, comparing also having the integrated fibrate as well as the flow cell. So that was really something to get there. But yeah, it's really the overall process also, as you mentioned, that is really needs to be ready for this and also be ready for the change. Because if you would look at how it was done like the last decades, I think there had been not so much change. And this is really something to kind of really dramatically, as we talked about Pharma 4.0, it is really something that is a shift in several ways, right? If it comes to workforce, if it comes to process, if it comes to analytics, I think all of them are really implicated in a similar way.
Okay. So, and I know earlier you were talking about regulatory. So what types of additional support/documentation did you need to implement the FlowVPX? And also, was there any pushback or comments from the FDA or EMA?
So the good thing is, as we were dealing with, as it's technology-wise, we're also calling it A280, but it's basically online A280. So hence there, from a technology perspective, it was quite similar. So what we always had to have was, how does it compare with the current offline technology? So usually we always had to have a certain data package to show that this actually is always the same. And we are really also doing this, as mentioned, for the monitoring phase, where we usually both methods at the same time and really have them there to really convince, to have this strong package to show that there is no big difference in using either technology. So this was, I guess, one of the main points we had to bring to basically have them convinced.
Okay, but I mean, ideally, eventually they were convinced. I'm just going through. I think this may be all the questions, and there might be one last more that I missed. Okay, oh, well, one last one. For companies that are planning to do something similar with your project, what piece of advice would you give them?
Also a good one. I would say to really see the stakeholders and employ the stakeholders early on in the process, right? I mean, it's as mentioned, so using also regulatory early on to already know what they want, what they basically plan to have. Includes people from the shop floor and engineering early on to have that. Also include QC, include analytics at an early stage to really see what hurdles and basically, yeah, roadblocks you might face over time. And I think this is really something that, if I would need to give this along for the time that we have working on all this technology and things, I think this is really something to give along to involve people early on, to really know what kind of questions might come up and might pop up, and which things to tackle.
Okay. I know I said that was the last one, but people keep shooting more questions in. So did you test it with the high protein concentrations? Like, say, if so, how high did you guys go?
Yes, so we also did this, especially in the drug substance space. For us, we even tested it above, I think, 300 mg per ml. So this was really kind of one of the highest one we tested it, and also over-concentrated material we had. Yeah, let's see where things will go in future, if we even might end up with even higher concentrations or so. But for now, that is basically one of the ones we had.
Yeah, and I mean, I would like to add to this question too, just because I am also a user of the FlowVPX technology. I've tested it all the way up to, like you said, a little over 300. I think 400 mg per mil was the highest I was able to get, with an extinction coefficient around 1.5. So of course, that's always going to matter in your process. You might be working one with higher or lower extinction coefficient, but yeah, that sounds about right. Okay. Let's see if give someone one more minute. If another one's going to come in. Don't think so. So I just want to again thank Marius and Renata so much for joining. This concludes our webinar. Thank you everyone for attending. It was nice to have you guys here. Thank you so much.
Thank you.
Thank you.